Welcome to Sekisui XenoTech's first presentation of the ADME 101 series, a comprehensive new collection of educational short videos to serve as a resource for researchers and industry professionals to navigate ADME, DMPK, and DDI within the complex landscape of drug development. As we continue to update and add to our repository of resources, we welcome questions and requests for topics our experts may address in future presentations. Please visit the link below to request a topic, submit feedback, or get in touch with our team. To kick off our series, we are pleased to introduce Dr. Joanna Barbara, the Vice President of Scientific Operations here at Sekisui XenoTech. Joanna obtained her Ph.D. in analytical chemistry from the University of Florida. She has authored or co-authored numerous scientific posters and papers, and regularly represents Sekisui XenoTech as an invited speaker at various analytical and drug metabolism conferences. Joanna has extensive experience in DMPK, regulatory compliance, and process and project management. As Vice President of Scientific Operations, she is responsible for the development, design, operation, and improvement of Sekisui XenoTech's scientific division.
Thank you, Madison. We're going to talk today about main aims of drug development and how various acronyms fit into that. We're going to focus on DMPK, drug metabolism and pharmacokinetics, and then talk about how that's based on ADME properties and how it relates to DDI a little bit. At the very highest level, the main aim of therapeutic drug development is to get a compound that does something helpful to a disease state into the form of a medicine that we can dose to patients. Obviously, that has to be efficacious. It has to be able to do its job, but we also have to make sure it's safe for people to take. And one of the biggest challenges is getting from a chemical compound that does something into an amount that's pure enough to be able to dose to patients, and then into some kind of dosage form that we can deliver to patients. So usually the beginning, and this is the screening portion of drug development, is looking for some biological target that relates to a disease state. And it's generally a cellular structure or a molecule that, if you can find a compound to interact with it, will change the disease state in some way. But the problem with that is that you can't just give a chemical compound to a person and expect them not to experience toxicity. So we have to get it to be able to be appropriate for humans to be dosed with, to reach the site of action and to do its job, and then be eliminated from the body because it's not meant to be there. It's technically a poison. So DMPK science explores and explains how all of those things can happen. An example I like to use is a tyrosine kinase example in cancer therapy, because unfortunately most people have some experience with cancer and understand it a little bit. So tyrosine kinases are enzymes involved in cell signaling to stimulate division. And if you look at the top picture up here, what usually will happen is that a growth factor attaches to the tyrosine kinase, which is the little yellow guy, and it binds to a receptor. And then once that happens, that sets off a signaling cascade that tells the cell to divide into two. It's important in cancer therapy because tumor growth is basically unwanted, undesired cell proliferation. So you're getting a lot of cell division and it results in a tumor. And so one of the important therapies that we use for cancer prevention or cancer treatment is to inhibit those tyrosine kinases so that we can prevent the cell signaling and stop the cell division. And in this scenario, if you look at the bottom example, what happens is that when the growth factor binds to the tyrosine kinase, some other compound is getting in the way and interacting with that tyrosine kinase to stop the signal from happening. So the nucleus doesn't receive the cell signaling and there is no cell division. And it's very effective. It's a common drug class, but the inhibitor compounds have to be able to get into the cells in order to interact with the receptors. So we're going to go back to our acronyms for a minute, and we're talking about DMPK today. The DM is drug metabolism, and we'll come back to that in a little bit, but for right now, we need to cover some vocab. Drug metabolism just means chemical conversion of the drug into other related compounds. And it's usually caused by drug metabolizing enzymes, or DMEs, so proteins that will interact with the drugs and turn them into something slightly different. And the something slightly different is metabolites. It very often happens in the liver, and so we typically will talk about liver metabolism. And then PK is pharmacokinetics, which sometimes is broadly defined as what the body does to the drug. And the stereotypical picture that we show and we talk about when we're talking about PK is this plasma concentration time curve This is the figure that shows us concentration of drug in plasma or in blood, usually plasma, from the time when the patient or the animal is dosed until the time when the drug is cleared from the organism. The target here is to try and get the drug to reach this concentration range, the therapeutic range, which is the range at which the drug will do whatever it is to impact the disease state. And so some of the things we're going to talk about in a minute are shown in this curve. This is the absorption phase. This is arguably the distribution phase, and then elimination happens over here because we need to be able to clear the drug out of the body. That all happens because of ADME. Drugs need certain characteristics to be able to be used for therapeutic benefit. And so we evaluate the compounds for ADME properties. This is the list of the ADME properties. Sometimes people include liberation and toxicology in these, but we're going to focus on the main four, which are absorption, distribution, metabolism, and excretion. And compounds that are druggable, i.e. they can be converted into real drugs, have appropriate ADME properties to help make sure that they can become viable medicines at the end. Some of the other language that we use here, I'm going to define, is disposition, which basically means what happens when you combine all of those properties, and elimination, which is a combination of metabolism and excretion. Sometimes the excretion and elimination terms get thrown around and used interchangeably. So the first step is absorption, and this is particularly important with oral drugs. We like to develop drugs that can be dosed orally because it's convenient to be able to give to patients to take home and self-dose. So oral drugs are very desirable. But what that means is that they're going to be delivered into the GI tract, and they have to make it from to the bloodstream. Biological barriers like the skin, like cell membranes, are meant to keep foreign substances away from the cells and away from sensitive organs. We have to be able to get the compound to traverse those barriers, and it's impacted by various characteristics like solubility or molecular size. Sometimes we can have passive permeability, so sometimes drugs are very small, and they're appropriate at biological pH to just be able to move through the intestinal wall, for example. But in general, a lot of drugs are larger than that and do not naturally just traverse that wall, and so that's when transporter science comes into play. We're not going to talk about that, but there are proteins that actively move the drug through those barriers. Intravenous administration is sort of the ideal scenario and also a not ideal scenario. It's ideal because if we introduce a drug intravenously, we know that the vast majority of that compound is going to go directly into the bloodstream. There is no absorption phase, and we know plenty is available to do its job, but it's very expensive and undesirable for a patient to have to go to the hospital and have an intravenous infusion every time they need a dose. So mostly, as I said, people want to develop oral drugs. They have to traverse the gut and the liver to make it to the bloodstream. And the way that happens is when you ingest the drug, it goes into the stomach. Some is absorbed at that time, but most of it's going to make it to the intestine. And so the way that it works, the way that absorption works, is usually it's going to move from the intestine into the portal vein and eventually go to the liver. And then the liver is going to be responsible for moving it into the systemic circulation. And the other complicating factor here is hepatic metabolism or liver metabolism. But if we don't have that absorption phase, then all of the material is just going to be excreted in feces. And we have to make sure it gets to the body. So the next step is distribution. Once it's in the bloodstream, the drug has to be able to move to the site of action. If we think about the cancer example again, then we know that we have to be able to get the drug to the tumor cells. So this is important because if it doesn't make it to its site of action, then there isn't going to be any therapeutic benefit. There are various considerations in this, particularly vascularity. If the blood vessels aren't going to go there, then it's not going to get there. We also have to be thinking about transport again, because now we have to get things from the bloodstream into cells. Can be passive permeability or some active transport. And then there are things like the blood-brain barrier or placental barriers that are again trying to keep the drugs out of various sensitive structures. And so those are part of this. And plasma binding proteins are also part of this scenario because there are proteins in plasma that are going to bind to drugs potentially and reduce the amount that then can be distributed to the site of action. Assuming we get to the liver, then metabolism is going to happen. It isn't just the liver. Sometimes it happens in intestines and kidneys. Multiple organs have drug metabolizing enzymes in. that one of the liver's main functions is to biochemically change a drug or foreign substance to make it more hydrophilic. Most drugs are quite greasy, and in order for us to excrete them in things like urine, which is basically water, we have to make them more water soluble. And so the job of the drug metabolizing enzymes is essentially to take things like the little greasy CH3 group on this ibuprofen, change it in some way to make it a little bit more hydrophilic so that we can excrete it and get rid of the drug product or metabolites. In this case, the ibuprofen is hydroxylated right here at this greasy hydrocarbon, and then it's further oxidized to make it even more hydrophilic, and then the carboxylic acid metabolite is excreted. It doesn't just happen at this one position. It can happen at lots of greasy places, just like this other carbon group. So we get omega -1 hydroxylation here, and these are the main metabolites and will be excreted along with whatever is remaining of the ibuprofen through the excretion route. Excretion is defined as the irreversible loss of drug from the system. It has to be gone. It has to be gone completely, as in it cannot return to the site of action or to the bloodstream. We have to be able to clear drug-related material, which is drug and metabolites, from the organism to prevent toxicity. If it stays, we'll just build up and end up having some adverse effects. It commonly occurs through the kidneys, so we have renal excretion that way in urine, or through the liver, which is biliary excretion for bile and feces, or through the lungs, which is respiratory excretion through breath. But there are other mechanisms too. We sometimes excrete things in sweat or tears or saliva. And once we've completed that, that's the end of the ADME cycle. The other things we did touch on are liberation and toxicity. Sometimes people will say LADME or LADME tox. Liberation means the release of the active substance, so that specific compound from the delivery system. A tablet has the active substance in it, but it also has a lot of excipients. So various processes have to happen to release the compound from that tablet, and that is classed in liberation. It's part of the absorption consideration too. Then toxicity of a drug is about whatever side effects or adverse reactions we might have as a result of the drug's presence. They're both important considerations for safety and for efficacy, so we need to make sure that we consider those as we're developing the drugs. So compounds that have desirable ADME properties and will ultimately become usable drugs get into the bloodstream. They can be absorbed. They move from the bloodstream to the site of action. They're distributed appropriately. They remain unchanged long enough to be active, or they're converted to safe, maybe active metabolites, and then they're completely excreted in a normal excretion route, and hopefully don't cause harm to the organelle system at the concentrations needed for therapeutic benefit. We're going to talk for a moment about drug-drug interactions, but this is a topic for another day. Drug-drug interactions occur when a drug causes changes in the PK or ADME behaviors of another drug that is in the system at the same time. And it's usually caused by changing available levels or activity of either a DME or a drug transporter. And so what can happen is maybe you're clearing the drug in a different way now. So you end up with either decreased action of the drugs, it's not as efficacious as it used to be, or sometimes you can end up with it being more efficacious, and so more of the effect happens than you were intending. And then the third problem is if it causes adverse effects. So these are important considerations, and they often derive from the ADME behaviors of the compounds in question. So overall, we talked about the fact that drug development is aiming to turn a compound that has an impact on disease into a useful, viable, practical therapy, and that DMPK and ADME properties determine how that will happen. They can also help predict DDI potential, but as I said, that's a topic for another day.
Thank you, Joanna. If you have any questions regarding Joanna's presentation or requests for future ADME 101 topics, please feel free to use the link on your screen. Sekisui XenoTech has over 25 years of experience in drug metabolism and DDI research. Our extensive selection of products and services can be explored on our website, and if you'd like to speak with a representative about your in vitro or in vivo non-clinical needs, you can use the form at the link below and a representative will reach out to you. Thank you for watching, and be sure to keep your eye on our social media and newsletters for upcoming presentations.